So I was just saying that next month, Connie McKinney will be talking about her book, The Arden School, complete with her interviews that she's done. That's October 18th. Tonight we have Dr. James Schwaber, who moved to Arden in 1981 with his wife, Mary Brent Whipple. Many of you know both Jim and Mary Brent from outside of here, but he came here to join DuPont Company's expansion into the life sciences. Currently, Jim serves as director of the Daniel Bow Institute for Functional Genomics and Computational Biology at Jefferson Medical School. And he is the endowed named Daniel Bow Professor. His research focuses on the emotional visceral neuraxis, extending from forebrain structures, especially limbic forebrain to visceral organs, in particular the heart. I had to read that because, you know. And in his talk tonight, he's going to be talking about the vagus nerve, and he's going to sketch an overview of the anatomy, the history, the present state of research, and including the ambitious current National Institutes of Health SPARC project. I don't know, SPARC project that seeks specific organ effects from nerve stimulation. And I've got, before I turn it over to Jim, we may have a little technological glitch, so bear with us because we're trying to do in live and Zoom presentations here. But I had not heard of the vagus nerve till recently, unlike many other people. Every time I mention it, somebody, oh, I want to hear that. Oh my God, that should be so interesting. So I think this is where popular culture and science come together. And I found out that TikTok has had over 64 million views of their videos on the vagus nerve, and Instagram has had over 70,000 posts. So this is very popular. People are trying self-stimulation and also looking for external stimulation. So we'll turn to Jim to separate fact from fiction and give us some information. Please welcome him.
You fixed it.
I guess I need to speak in this so that the Zoom works, right? Hello. I just want to clarify one thing. When Mary Brent and I moved here, we moved from Burlington, Vermont. Vermont is pretty different than Delaware. It's not suburbia. And we were so delighted to discover Arden. It seemed like Vermont. And, and so in some way it sort of is. So happy to be here. I'm going to give you a informal talk and a formal talk. Two. So this part is the informal talk. I'm just going to free associate around on topics that I thought you might like to know about, that I maybe know a little bit about. And we can, just see what's interesting. And then, so at the bottom, in the last little gizmo there, I'm going to switch over to a formal talk that was given last week, by my colleague Raj in Australia to an international meeting on this subject area in Cairns, which is on the Great Barrier Reef. And he's just back, a little wiped out, but excited. And so I'll show you his talk and you can see what it is I actually work on. I'll try to keep it light, just say this is about this is about that, but you just get a flavor of the kind of stuff we're trying to do. Right, that's the plan. So looking at the topics here, the first one is there's a lot of interest in the fact that when you electrically blast the vagus nerve with stimulation, that you get effects on brain function.
Don't touch.
A lot of effects on brain function. The more things you look at
Yeah.
in the brain, the more things get affected. And I'm going to
Anyone say
read
one?
you a little vignette
Yeah.
of a depressed woman and her experience with vagal nerve stimulation as a narrative to get you engaged. I
Yeah.
believe that narratives are crucial to,
What do you want to
us
play
to
with?
care about it. So I'll give you a little narrative. And then I'm going to say, that's not all. That's where we are now. Where is it going? Let me show you a few of the things that are happening, which is quite remarkable in terms of the ability to maybe stimulate much more specifically, get much more specific effects. We ourselves have done some work on the vagus nerve's role in withdrawal from alcohol. When people go through withdrawal from alcohol, from opiates, a lot of that effect is actually mediated by the vagus nerve, and modulating that vagal effect could make it easier for people to get off the drugs. So we'll talk about that a little bit. Okay, there's a lot of mystical stuff out there about the vagus nerve, meditation, so-called polyvagal theory, and the opposite of that, where people get very stressed out and the emotional parts of their brain blast down into the vagal output, cause arrhythmias, and people die. That actually happens. People can drop dead and from stress.
Yeah. Am I on mute?
Touch on that. And then we'll go over to what I actually work on, which is the other thing, the mirror image of the effect of the vagus on the brain, rather the effect of the brain on the organs through the vagus. That's the two-way street. Got it? And then I'll go over and show you the slides that my colleague showed, and we'll just zoom through them. You're not gonna, I don't expect you to, we're not gonna drill into all the different parts. I just want to show you what the kind of stuff is we work on. FYI, we'll go as far as we go. When you get sick of it, we'll stop. And then as a special treat, the styrofoam box. In the styrofoam box is an actual human vagus nerve and an actual human heart. And Mary Brandt was just all over me that I had to also bring a human brain. So we could all lay it out there. Brain, nerve, heart, there they all are. I held, she's very mad at me, but I didn't bring the brain. Yeah. This better? I'll feel like a comedian. I'll start telling jokes. So anyway, I don't have the heart. I mean, I don't have the brain, but I do have the heart, and I do have the nerve, and we have the tray. I'm not going to put it out till the end. Why? It stinks. And many of you may not enjoy being exposed to it, but if you are curious about what an actual human, stuff looks like, it's in the box. And my dear colleague and friend, Dr. Leonard Eisenman, who's a superb anatomist, just in the last 6 or 8 months, has taken extensive branches of the vagus nerve out from human cadavers and the attached organs. And this is an example of that. And we're going to use that for a number of things, especially from a genetic point of view. I think that may be more clear as we talk about what it's for, but I'm going to hold off putting that out till we're done here so you can flee. Okay, so that's the first part of the talk. Okay, here's the part I'm going to read to you. I'm not going to read to you the yellow highlighted part. You have to read that on your own if you want, but here's the narrative. On a recent Friday morning, a gray-haired woman, whom I will call Sally, arrived for an appointment with Carl Diceroth, a psychiatrist and a neuroscientist at Stanford. Sally, in her 60s, suffered since childhood from a major depression, had tried all the standard treatments, counseling, medication, even electroconvulsive therapy. Nothing helped. She had spent much of her adult life in bed and twice suffered attempted suicide. 7 years ago, she was referred to Diceroth, who uses a combination of unusual medications and brain stimulation to treat autism and severe depression. He accepts only patients for whom all other treatments have failed. On Diceroth's advice, a surgeon implanted beneath Sally's left collarbone a small battery-powered device that regularly sends bursts of electricity into the vagus nerve, which carries a signal into a deep brain structure doctors think regulates mood. Originally developed for epilepsy, vagus nerve stimulation, VNS, has been approved by the FDA for use in the kind of treatment-resistant depression from which Sally suffers. But it's not understood why. Sally says it's transformed her life, except from one period where she was going pancake, she had just experienced a few dips. She was not mollified. Mood's been down, just spiraling down. No suicidal thoughts? No. With sudden urgency, she asked to have the VNS current increased. Will you please go up to 1.5? She'd been receiving 1.2 milliamps every 5 minutes for 30 seconds, but that wasn't able to feel the effects. You're tolerating the device well. I think we can go up. He handed her a programming wand, which looked a little like a Y, gaming remote. She placed the broad, flat end against her left collarbone and over the implant. Diceroth took from his desk what appeared to be a smartphone. He thumbed the screen. The wand emitted a trilling tone. "I can feel it," she said. In the course of the next few minutes, Sally underwent a remarkable change. Her frown disappeared and she became cheerful, describing the pleasure she'd had during the past Christmas holiday. She was still smiling and talking when the session ended. Diceroth walked her out to the reception area. How's that for a story? It should all be so smooth. We'll talk about inconsistency and how some people have a wonderful response and some don't, and why, and what we're trying to do about that. Yeah. Oh, yeah. Is this working? Can you hear? Hello. The future, what's the future? So you got this far by just blasting on the nerve. What's next? What else is possible? What might be able to be done? Let's look at a couple of things that are kind of cool. One is, so it so happens, I don't have a pointer. You have to just look and see that on the right side, you see the nodose ganglion. That has the cell bodies. So it so happens that the vagus nerve sensory fibers, their cell bodies are not in the brain. They're in the periphery, inside the nerve, pretty close to the brain, but not in the brain, in a blob called the nodose ganglion. And they're all in there. And here they are from all the different organs. And it turns out that each one of the organs has a distinct set of molecular profiles, distinct set of genes that they express that subserves the function that they do. Cool. Now, it so happens that Diceroth, the same shrink, is super famous for having developed a way to stimulate selectively genetically distinct populations with light. That's what he wants to do. Blue light for these guys, green light for these guys, wake them up and get very selective effects into the brain without electrical stimulation. That's what's on the horizon. And that's going to be very amazing. And here's a trick for you. It turns out there's a branch of the vagus nerve. So the main part of the vagus is a big trunk that goes down through your cervical region, thoracic region, into the gut, all the way down. But it sends a branch called the auricular branch, curls around into your ear. And it innervates the part of the ear, not the external pinna, the internal. So the ear canal, all around in here, you can just sit and stimulate the heck out of it. That's the auricular branch. Doesn't need any fancy electronics. You could do it. You could have a friend give you an ear massage. And it could have a profound calming effect. Worth a try. Yeah. And one last thing that on the horizon, I'm giving you horizons here. Oh, wait, I've got my slides are out of order. Sorry, I'll get to it in a bit. All right, we're up to alcohol and opiate withdrawal. Yes, so I have this wonderful student who many of you have met because he's given Scholars Guild talks. His name is Sean O'Sullivan, extremely handsome young lad, now a resident in psychiatry. He worked with Carl Dijsselroth on all this at Stanford after he graduated. And so he has the idea that the reason people have such a hard time getting off drugs and off alcohol is because in withdrawal, the withdrawal process is so miserable. People just suffer. They feel like near death's door. And the reason for that is to a large extent due to the gut. There are a lot more mu-opioid receptors in the gut than there are in the brain. And the alcohol effects in the periphery are profound. When you withdraw alcohol, the whole gut seizes up. And then that reports back to the brain. You can see here on the left side, the stuff is coming up from the gut into the brain, and then up to the parts of the brain that are involved with emotional state. And there it's appreciated as misery. And so his question is, could you interrupt that flow of information either in the so-called NTS, where he's got antagonists to the transmitter, or in the amygdala, where you experience the anxiety as it's an inflammatory process, and you can interrupt that. And by interrupting either of those things in people experiencing withdrawal, the withdrawal symptoms become much ameliorated, much easier to deal with, much less pressure to get back on the alcohol or the drug. So that's that. Pretty good, huh? Good for Sean. And here's the one I was thinking I had before. This is so funny. I mean, it's just funny, like, are you kidding me? So a group just published in the last couple of months, a group of investigators at GE and Yale. So organs have a part of the organ that's dedicated to the input output, where the blood vessels go in and out, where the nerves go in and out. It's not like random all over the organ. It's just one spot. And that's called the hilus or the hilum. There's this thing called ultrasound. And there's this thing called focused ultrasound, where you can put as much energy as you want to into a very local spot. And this group focused ultrasound on the hilum of the liver in patients who were diabetic and obese. Diabetes went away. Oh, it was pretty sustained. It was like several hours of stimulation, but once. Done. Diabetes went away, they lost weight. So far, so good. But I thought you'd find that entertaining. I do. Okay, so now we're down to the part I actually care about. I don't work on the input, I work on the output mostly, except for this alcohol stuff Sean did. Alcohol and opiate. So the vagus nerve also has an output effect, a mirror image to the input that affects the state of the brain. There's a brain effect that goes out and affects the organs. And so let's talk a bit about that. And here it is. I'll read it to you. Experimental data have demonstrated that stimulation of the vagus nerve is able to reverse ventricular remodeling of the failing heart. There's also evidence that increasing parasympathetic activity may stimulate the production of nitric oxide, reduce the devastating inflammatory processes involved in heart failure. Good stuff, huh? And I also want to tell you about another thing you can do at home, RSA, which is respiratory sinus arrhythmia. I'll tell you about that in a minute. Another trick you can do at home, not just stimulate your ear, but another trick. And so let's look at the stuff. Here's the bottom of the brain, looking at the brain. from the ventral surface. And what you can see here, the brain is encased in the cranial vault. It's isolated from the world. Has no idea what's going on, except for the cranial nerves. There are 12 cranial nerves that convey information about the world from the brain to the outside, from the outside to the brain. And you can see them, they're ticked off here: olfactory, optic, oculomotor. Get down to 10. That one down there, that's our vagus. This is the thing that I always teach with that all the students love. Get it? Is that fun? So you're not going to learn the cranial nerves tonight, but that's what they are. Okay. And then here's the nerve. Vagus nerve heads out from the brainstem. It charges out of the cranial vault. So the cranial vault is protected. It's bone everywhere except for these little holes called foramen. And that's where the nerves and blood vessels go in and out. So the vagus goes out through something called jugular foramen. And it gets out and it forms a nerve. And then it trucks on down to talk to all the organs. That's the vagus. Good. Human vagus nerve. We have one. There he is. He's a male, human male, cadaver number, ready to rock and roll. If you can stand it. It could be a challenge. So that's the anatomy. This is what it looks like. If you don't want to see it in person, there's some pictures of it. And on the left side, you can see the trajectory of it coming out from the jugular foramen, forming up and going down the entire cervical length until it branches down at the bottom of that image. You can see 3 branches coming off. One of those branches is called the laryngeal or recurrent laryngeal. That goes up to your larynx and pharynx and is controlling my ability to speak right now. The second one goes to the lungs and controls the volume of the lungs and the expansion and contraction of the lungs with breathing. And the third one goes to the heart, and that's the one that we study. Now, this is why everything is so hard. This image is an electron micrograph of a part of the vagus nerve. Okay, and you can see everything in it that makes life hard. This micrograph was made by Mary Brent Whipple. She was the EM tech in our department when I had a project to look at the vagus nerve, and she panicked me that she would screw it up, but she didn't. She got the best, by far the best microscopy of the vagus nerve to date. And this is long before she gave up. This woman frustrated me. She had tremendous technical skills and produced images like that. And medical school was beckoning her. All her friends were in medical school. She should have just gone to medical school. Nope, I want to be a social worker. So there you go. And but what you see here is the problem. These big dark circled things, that's called myelin. It allows conduction down the nerve to go. So these are all nerves. These are all axons of neurons, and they're conducting information from brain to the organs or from the organs to the brain. And the ones that are big myelinated guys are probably afferents, sensory. They're powerful, they're fast. And the ones that are little tiny guys are unmyelinated and they're slow. And a lot of them probably are motor. It's not important for them to get there so quick to the gut or to the heart. It can be slow. and the intermediate ones are in between. So you have all these different things, and they're tremendously different in their sensitivity to stimulation. You go to electrically stimulate them, the big guys go off quick. Little guys are hard to get go off. How do you control the stimulation to get the ones that you want? Right? This is a cross section through the nerve. So this is just a piece of it. In those days, we didn't have the electronic means to paste it all together into a whole nerve. So this is just a piece of it. This is ancient history. We were kids. Oh, well, there are lots of branches. When the nerve first emerges out the jugular foramen, it gives off the auricular branch. Very shortly thereafter, it gives off the superior laryngeal branch that goes to the larynx and pharynx, and so on. There's a bunch of branches that are right here. And then as it doesn't do too much branching as you go down until you get to the three I just showed you, where you get the laryngeal, the respiratory, and the cardiac all coming off. And at that point, the remainder of the nerve plunges towards the gut, goes through the diaphragm and innervates the gut. And then you get, this, all the different gut branches, which I could go into if you really want to know. So where were we before I got lost? Where was I going with this? So it's really hard. It's the challenge is how to produce, stimulation. Which fibers are the ones you want to stimulate, and how can you selectively stimulate them? Right now, it's just a crapshoot. You just stimulate and hope you get something, and that's why it's so variable. Sometimes you get what you want, sometimes you don't. Just depends. And I'm going to tell you about the Spark program that aims to fix all that and what we're up against in it, how it's working. Okay. MB. All praise to her. That is an amazing micrograph. We did that in 1970. That was a big shock to see the variability of it. It was like, oh my God. How in the hell are you going to pull out the ones you want from that? And what makes Mary Brent ready to murder me is I used to have whole boxes of gorgeous micrographs like that for the whole nerve, and I lost them. It's only been 50 years. I mean, a lot of moves. I want to kill myself too, but
This guy's an asshole.
anyway.
What a
Okay,
freak.
so life in the fast lane, life in the slow lane. So we're going to talk just about the vagus nerve's
That's it.
effects on the heart. Okay, and it's vagus nerve effects on the heart arise from two structures in the brainstem, as you can see on the left there, the NTS and the DMV, and they go to different parts of the heart. Now, important information. The vagus nerve does not innervate anything specifically, only innervates neurons. All of the vagus nerve output only innervates neurons that then innervate the organ. So it's really important to know where the neurons are in a particular organ and how they're organized and what they do, and then understand the inputs to them from the vagus nerve that will explain how
I'm
the
glad
vagus
she's
works.
back at the gym.
It's not the direct effect of the vagus on the organ. And so there are these two outputs, one from the nucleus ambiguous, one from the dorsomotor nucleus, and they have very different effects. One is very fast,
Is there music? What the hell?
nucleus ambiguous, and has detailed regulation.
This
of
is
the
so
beat
boring,
of
I
the
think I'm
heart
gonna
from
throw.
beat to beat, really tight. The shape of the
Nuh-uh,
beat,
you
the
actually-
rate of the beat, how frequently the beat is, all that stuff is tightly regulated by the brain. Then you have the dorsomotor nucleus, which is slow. It doesn't do that much. It doesn't have that tight control of a rate. the nucleus ambiguous output. So if you stimulate the vagus nerve electrically, you wake up the nucleus ambiguous fibers, you stop the heart cold. It just stops. Not another beat. Done. So you let it go. That's how profound the nucleus ambiguous effect is. It controls the heart. The dorsal motor nucleus, meh, not so much. It's doing a lot more to inflammation, modulation, metabolism, all these kinds of things that are more regulatory to the health of the heart. So there you are. I could go into details, but here is the fast lane. And here comes the practical advice to you, something called respiratory sinus arrhythmia, RSA. In healthy people, and you'll soon find out if you're healthy, there is a pretty heavy RSA in which, with every breath, the rate of the heart changes through the breath, just like you see here. Pretty cool, huh? In young, healthy, athletic people, looks like that. And old, sedentary people, maybe not so much. Breathing affects your heart. Yes. Absolutely. Use these mechanisms. Absolutely. So there are different, and I'll show you in a minute. I hope I'll show you I hope a model structure in which we're taking apart the basis of RSA. There's some of it that's at the heart itself. Some of it's due to the carotid sinus reading the blood gases. Some of it's from lung stretch. There are different influences that drive this. But this is the output. You get this arrhythmia, which was thought to be unhealthy arrhythmia, but no, turns out metronomic beating along of the heart is not healthy. My dear friend and former student, Julian Payton, has a little company that he's in Auckland, New Zealand, and they've got a little company that has a novel pacemaker that imposes RSA on the heart. It reads respiration, stimulates the vagus, and causes it to speed up and slow down, as if RSA were fully robust and healthy in people who are in heart failure, whereas obviously not. And he's getting tremendous effects on heart failure, big increases in cardiac output. So it's an important phenomenon, and you can measure it at home. Another little trick, just like the auricular, some take-home messages. So it's why some people have got rhythm. Russ, I believe you.
I'm not sure, Zoom people can hear the questions, so if, right now, if you could repeat them, and then later after the presentation, we'll take more questions with the mic.
Okay.
Yeah, thanks.
So this is the slow lane. And the slow lane is more complicated. It's an output from the dorsal motor nucleus, but it wakes up all these modulatory factors that regulate neural function, that regulate metabolism, that regulate stuff that we have to investigate. And that's stuff that counts for heart health, long-term heart health. We're just beginning to pick that apart, as you'll see in the next bit. Okay. Now we're ready to go to my talk. Here's where it's going to speed up and get ugly. Okay. I hope. Done with that. End of slideshow. Go away. All right, so this talk was presented by my colleague Raj Vadakapali. I don't want to record. Stop it. Okay. There we go. So, I organized a symposium together with Julian, aforementioned Julian, who's in Auckland, to occur in Cairns last week. I was the co-organizer who was absent, but I didn't go. Bum. But Raj went and he gave a talk. He was on the program and he did it. And this is his talk. You can see the kind of stuff we do and worry about in detail, much more detail than you want to know, but at least I'll fly over it and you'll get a flavor, okay? That's the idea. So, okay, so here's the team, all the different people that are involved. And the funding sources. I have to write grants constantly. It's my joy. And on the right is something that emerges from a joke that I came across a long time ago and that I really like. Bertrand Russell was giving a talk, an address to a club much like this. And he was talking about the heavens and the earth as a sphere rotating about the sun and so on. And a lady in the front row rose up, waved her umbrella in the air and said, "Oh, that's all very well, young man. But everybody knows that the earth is a giant platter resting on the back of a giant tortoise." And Bertrand Russell thought about that for a minute and he said, "Well, very well, madam, you may well be right. But in that case, upon what does the tortoise rest?" And her response was, "Young man, this tortoise is all the way down." So that's the joke. And so this there in science, there are many turtle things like this playing off that joke that are taking, making fun of the whole thing. And we do it because we're guilty of the same thing. We're all about networks. We don't think there are point source causes of anything. We think everything is a network interaction. It's a process. It occurs in time. And that's just our mantra. And networks all the way down. So we're making fun of ourselves. You can read this. I don't know if any of you have heart failure, but you well might. Your dad did, right, Hugh? It's a scary thing. We are control of the heart, fast and slow. Again. And so now we're going to talk about Spark a little bit. The NIH, in its infinite wisdom, has recognized the importance of this and reared up and created a program called Stimulating Peripheral Conditions to Remediate, Activity to Remediate Conditions, Spark. And it started in 2015, 2016. We've been privileged to be an important part of Spark, of course. And it's been a marvelous program. It's a team organized. It's many different groups all interacting. We get together a couple of times a year. And it's been marvelous. And it's really going. And it's trying to figure this out. What to do about that diversity of axon types and sizes, and how can you begin to have a selective effects that are therapeutic. Spark is not interested in the input to the brain and effects on mood or anything like that. It's only interested in effects on organ health. And our part of it is heart. So this is Spark, and its vagus nerve is even fancier than the one you saw before. And map of the peripheral nervous system that Spark is studying. Life in the fast lane, health in the slow lane. Heard this before, right? So we're going to go with the takeaway concepts that Raj stuck in and try to get nothing more than that out of this. So the little brain at the heart. These are the neurons at the heart that are the target of the vagal inputs. The vagus nerve goes to the heart, but when it gets there, it talks to the intrinsic cardiac nervous system, the little brain at the heart. And we have mapped that. And we know where it is, we know what it is, we know what innervates it and how. And we're begin to learn how it changes, how it adapts to injury, and how it adapts to things that can improve its health. So that's our game. Now, here's some recent publications last year or so on it, and some recent methodologies. And then it shows the team that we're part of, people in University of Central Florida, people at UCLA, people in Vermont, us, all interacting. And this is the ICN. This is the intrinsic cardiac nervous system in context of the heart. All right, have I gone too long?
No, you're doing great. You're doing fine. But let me share this over the Zoom. The question was asked. Ann was wondering if this is similar to how fetal non-stress tests look for variability in the fetal heart rate as a sign of a healthy fetus.
Absolutely. This is, it's bad in a fetus. You don't want your regular heart rate. But once you're breathing, that's when you want it. And in the case of the fetus, it's not going to be there because they're not breathing. And I think it's about 6 months by the time these reflexes kick in and you start getting these kinds of effects. So you wouldn't expect it in the newborn. So this is it, guys. Those are neurons in the heart. And they're the target of the vagus nerve. All the effects of the vagus nerve are through those neurons. And we mapped them. We know where they are. We got them. We did. We done good. So I'm not going to run through all of this. You can just look at how pretty the pictures are. I think they're pretty anyway. Oh, wait till you see the next one. This to me is the prize. 7 rotating hearts. See, turns out the ICN is quite similar across individuals and quite similar between male and female, except females have fewer neurons. At least, young females. There's a trajectory of what goes on with the heart with the female, premenopausal, very protected, not much danger of heart disease, postmenopausal, just like men, just as bad. So probably these things transition with time. I think there's a slide here that has some of that on it. It was amazing. Amazing. So male versus female. With the group in Auckland, New Zealand, we built a scaffold that we can compare across species and so on. And we have, as you'll see in a minute, a whole bunch of data from hog. This is about the genomics, the transcriptional profiling, that is involved with the different subpopulations of neurons. And here are 2. So now we're in pig. See up here in the right-hand corner? See the pig? If you want an experience, which I have had the horror of having, go on a surgery with a pig. These are 350 pound hogs, Newcastle hogs, splayed out on their back with their chest open and their heart sitting there and people doing things. And a bunch of cardiologists running around doing this and that. And that does make your hair stand on end. So, so this is pig and compared to mouse and human. And so now it gets neuromodulatory networks from different parts of the ICN. And here you get the connectivity. You see the fibers coming in from the vagus and just wrapping themselves around our ICN neurons. And now you see the two channels. The inputs from the nucleus ambiguous innervate neurons that express one type of acetylcholine receptor called nicotinic, which is very fast. And the dorsal motor nucleus input innervates neurons that express muscarinic receptors, which are slower and influence modulatory factors. So that's what these slides are about. I think, yes. No, that's not it. So these are all just, bioinformatic analyses that cluster transcriptional behavior of neurons into different groups depending on where they are. This is something called Visium, which is really weird, really interesting. There's a new technology by which you can lay down a tissue section of the brain or of the heart onto a slide that is prepared with a grid in which there's the chemistry for sequencing on this grid. So you can get every 20 microns, you get a distinct readout of the transcriptional expression that's going on in that part of the tissue section. So you wind up with this kind of, see it here? Can you see it on the bottom left? You can see the little, that's the grid. Each one of those is a distinct readout of the transcriptional behavior. Quite remarkable technology, and it's getting only better, much better. So that's fun. And so you can cluster the transcriptional behavior and begin to pull out clusters that are distinct for different functions and different connections and different regions. And that's what we do. And here's the takeaway concepts: 3D spatial, single cell, high throughput, omics. Now, how do we look at plasticity in the system to figure out where to intervene to promote cardioprotection? Now, this is interesting. Here's a really bizarre phenomenon called remote ischemic preconditioning. If you occlude the blood flow to a limb or whatever, just occlude blood flow to something for 15 minutes and release it. And if you do it repeatedly, even better, but once is enough. And then you injure, in this case, the heart, cause a heart attack or cause a stroke or any other damage. It's protected. Doesn't get it injured. Somehow the preconditioning, this ischemic event, releases things that prevent damage from occurring. It happens with the heart. It's totally dependent on the dorsal motor nucleus. And you can emulate it in this slide. You can see that blue light in the upper right-hand corner. That's optogenetics. Somebody's stimulating the dorsal motor nucleus with blue light, waking it up and producing ischemic preconditioning that prevents the heart from being damaged by a heart attack. And you can see it here in these, which I don't have a pointer, but you're supposed to be able to detect that when ischemic preconditioning precedes the heart attack, there's no damage. When it doesn't, there's massive damage. The damage is done. So if you do something like, in our case, we tied off the LAD, the left anterior descending artery that feeds the left ventricle, and killed the muscle because it didn't have any blood supply. But if you preceded that damage with ischemic preconditioning, it didn't hurt it. It was fine. as you saw on those previous slides. How? Good question. That's what we want to find out. And so you can't go around ischemic preconditioning all the time in case somebody gets a heart attack. But what you could find out is what are the protective factors and try to, in one way or the other, emulate them, either pharmaceutically, genetically. We're interested in genetically. So, oh, here's Visium here. You see, that's Visium. And 3D spatial transcriptomics. And look at here. Look at on the right panels, the distribution in 3D space, in the three-dimensional space of the neurons that Visium is picking up and how it's able to track them spatially. But that's a really powerful tool, we think. There's more Visium going past it, going past it. And so trying to define vagal neuronal types. Here we are, fast lane and slow lane. And Raj, being Indian, he had to warp my joke. That's it, folks. Oh, I'm sorry, here's more. This is our passion. There's this stuff called microRNA that you may or may not have heard of. There's mRNA, which is what is the intermediate between gene expression and protein expression. And there's other things called microRNAs that are small RNAs that don't make proteins, but what they do is they modulate the mRNA functions. And we think if we can identify the microRNAs involved in ischemic preconditioning, that would be a good target. And try to modulate them. Sir. Right. Okay, so the question is, what about diving animals? So there's a thing called the diving reflex. You know about that? So we all have that. Immerse your face in water, especially cold water, but any kind of water. An infant, you, me, and your heart rate plummets. Protect you from, needing to breathe. And so that reflex is potent, obviously, in dolphins and so on. It's probably an exaggeration of that reflex. It shuts off their breathing till they resurface. So this is a different mechanism. There are neurons at the heart, and the same neurons, they come from the nucleus ambiguus. And the same nucleus ambiguous neurons go both to the heart and to the lungs to mediate the diving reflex. And almost certainly those are the same guys that mediate what's going on with our mammalian friends who have to dive. Surely. Okay. Am we done here?
Well, not necessarily, but you can be. Okay, first question, Jim.
Modeling. Of course, we couldn't stop with just experiments. We do computational modeling, just like this is like climate, only it's heart. So it's all these different. How does the RSA emerge? How does diving reflex? And we want to explain it all by interactions within the elements in the model. So then you have this, and I think we're done. Thank you, Raj. Oh, he threw in some stuff from the Great Barrier Reef because that's where he was. Last week, lucky guy.
Here's the first question off the internet. Let's see. Scott asks, can you explain what happens in a vasovagal reaction, and can anything be done to avoid it?
Vasovagal reactions are the physician's favorite cop-out, right? You have no idea what's going on unless it's vasovagal. So nobody knows what it is. The idea is that somehow, vagal afferent feedback is reaching the circulatory system and shutting it down, and you're getting pain, you're getting sympathy as a consequence of the feedback from the periphery. That's vasovagal. But nobody measures it. It's a concept, right? You seem to know. I think, yeah. It's a physician cop out. I love it. We have to go to something when we don't know, right?
Okay, continuing. Ken asks, are there any predictions on what might be learned about the vagus nerve in 1 year, 5 years, and 10 years, etc.?
In 1 year, I hope we have some microRNA targets to manipulate to see if we can achieve some cardioprotection by manipulating microRNAs. In 5 years, I might be dead.
There are predictions out there probably, but we just don't know what they are, right?
5 years, I write grants and we put predictions and future directions and just make it up.
Prediction.
Make it sound good.
Do a third question here, if that's all right.
Yeah.
Let's see. Colleen asks, also, any current treatment options for anxiety or depression, or just studies going on?
Yeah, well, I read you the one with Diceroth with stimulation for depression. Mary Brent is the expert on this. I wish you were here instead of home with COVID, but my understanding from her is that anxiety is by far and away the rampant issue in mental health. And depression follows along with it to some extent, to some significant extent. And so, the vagal nerve stimulation has an effect on anxiety. And if you were able to, okay, here's the thing I didn't say. So in the Spark program, we're trying to sort out, is it possible to say selectively stimulate just those fibers that would ameliorate anxiety? Could you identify them and could you selectively stimulate them? And a year ago, I would have been optimistic. The data that's emerged in the last 6 months makes it look harder. It looks like within the vagal trunk, as you're going down the cervical vagus, there's a lot of mixing going on. Fascicles are forming, reforming, forming, reforming. I think that's how the branches are formed. That the little rootlets that come out from the brain are not functionally grouped specifically, and they have to get grouped en route before they exit as a branch. And so that makes it hard to discover them and selectively stimulate them en route, but nonetheless, you can at least identify, I think it looks like you can identify their size and character and try to, by the shape of your stimulating electrode, how it's oriented on the nerve, you may be able to improve your success rate quite a lot. So that's the current focus. Everybody hopes optogenetics, which I didn't explain to you, but it's the business of using light to selectively stimulate specific functional groups. That looks like a tough climb because you have to incorporate, channel proteins into the body of the nerve, and there isn't that much metabolism going on in the body of the nerve. So that's the challenge there. You have to get closer to where cell bodies are, where terminals are, where the metabolic action is in order to get these things incorporated. But we're working on it, guys. We're on it. The questions you're asking are the questions we're asking. Or I'll carry the microphone if I. Ischemic preconditioning. Okay, so if it turns out that ischemic preconditioning is the plasticity, it changes, right? The system changes as a consequence of your doing the ischemic event. What changes? But we think if a lot of the event is driven by microRNAs, we could jump in and emulate them. I don't want to get too technical, but it's possible to emulate, to block microRNAs or stimulate microRNAs selectively and specifically in particular parts of the brain. And we've done that. We cured hypertension doing that. There's an animal model called the spontaneously hypertensive rat that grows hypertensive between 6 and 12 weeks of age, and they're just fine. And then they're terribly hypertensive. We tracked what happens to their mRNAs and their microRNAs during the development of hypertension. And we looked at the microRNAs that were changing in parallel. We modeled it as bioinformatics analysis of how the mRNAs were being modulated by the microRNAs. Instead, if we went in and blocked the microRNAs, we blocked the mRNAs, we blocked the hypertension. Happened. We're going to have an offline discussion. This goes all the way back to cyanobacteria. Okay. There's a lady back here behind you. I can't hear you very well. Oh, you did? Don't you have high sympathetic drive? Okay. Oh, yeah. Okay. Yep, it's different. Well, no, that's not quite true, but they do have different effects. Seems to. If the trouble is, it's not absolutely consistent. You can't count on it. That's why it can't become like a marketable thing, because, 30%, 50%, it's not enough. Oh, the question is the ear stimulation profound enough to be clinically meaningful? Yes, it can be. Yeah, I didn't know that. Wow. The Society of the Ears. You're interrupting her. Who knows? Your guess is as good as mine. The question is, if you are trying to calm the anxiety, it is the humming that you do in meditation efficacious in part because it stimulates the afferents of the brain? Could be. How would you know? What's the study going to look like? Let's mount a clinical trial. Google is not experiments. Wait, your dad is next. Until you're tired, or the other person is tired who's doing it. But apparently there are devices. You know, I don't know what that would look like. It could get painful. Stop.
Other than stimulation, we talked about, the RSI.
RSA. Mm-hmm.
You said there was a home remedy or an exercise that you could use.
Yeah, it's called exercise. Yeah, yeah, just plain old. Yeah. Yep. Yes, sir. What's, I didn't get it quite. Say again. Yeah, sympathetic. High sympathetic drive. Yeah, there's a lot of talk about that, about, autonomic imbalance with low vagal tone and excess sympathetic drive. But I think that the sudden cardiac death is excess vagal drive. I think it's upsetting the rhythm. Driving the normal pacemaker away so that you begin to get, VTAC and off it goes. I think that's what happened. Sure. Yeah, no, it's not all interrupted because I just showed you all. Oh, the question is, in a heart transplant, do you lose all that? You lose the vagal input, but you don't lose the local circuitry. All the intrinsic cardiac nervous system is still working. I don't know if the vagus grows back in at all in a transplant. Maybe could. I don't know if anybody's even looked, but the intrinsic cardiac nervous system is still going fine. Yeah, and also little known, I didn't know this until I began to work with a transplant surgeon, but the transplant of a heart is not the whole heart, right? It's mostly the ventricles because that's where the damage is. That's the part that needs to be restored. If you try to transplant the whole heart with all those blood vessels and all that fragile, pin-walled atria and stuff, it'd be a nightmare. So that stuff stays, and that has the vagal innervation. Right? I didn't know that. And we wanted to get a transplant heart from a transplant guy at Jeff, and I thought we were going to get the whole heart.
Is this research being done worldwide in pockets all around the globe?
That's the meeting that this talk was from. Raj just did that. He was in Cairns, which is in northeastern Australia, near the Great Barrier Reef, at a meeting that is full of people talking about this stuff. Lots of fun for him. I didn't go. I stayed here and got COVID. I got to go to the Arden Fair and get sick. I should have gone to Australia. Oh, yeah. You guys ready for the box?
We want people to file up and look or what?
Let's see what's in here.
Hold on a minute, we're not quite done here. Thanks.
Yeah.
But I will.
Working with the wild. Well, that's pretty fun.
Yeah, yeah. There's more on either side.
I'm there right now, Mondays. I'm there for 11. So, the idea was to go to your. I'm out of your. Whoever teams go on the show. They give a few minutes and there's somebody else. I'm amazed what a kind of music there is.
Before we get all crazy here, let me, where's my phone? While Jim's preparing the specimen, let's just give him a big round of applause. Thank you.
Yeah. Yeah, well, there you go. Wait for the barbecue. You can see the vagus nerve on each side, and you can see it going into the heart.
Kind of hard to say.
Here's the nerve. Left and right nerve.
Oh, cool. Wait, what's on the?
It's harder in the heart. The vagus nerve,
In about a month.
heart, pericardium, or cardiac attack.
Oh, cool. Ow.
What were the dogs called where all the girls came together?
Have way more questions to ask.
Yeah, with the
There's an error and there's goes down. And all they do is subtract from 1 dollar. 2 weeks. It's cheesy. Well, I think in one area they're playing one way. I don't know. All I know is they're going to play all the way. This is third tour. And I'm hoping you'll be supposed to go to Europe in the new year.
I'm gonna go to the book. Let me.